So, welcome to week 5 interaction session for classics in neuroscience. Uh, Before I begin, does anyone have any doubts related to the week 5 video lectures that you would like me to address?
No, ma'am.
Okay, in that case, I will begin by first addressing some of the previous year assignment questions, and then we can discuss some of the important concepts that were discussed in this week's video lectures. So first question, what problem did Lars Lexell address during the war related to the experiments of Jack Eccles and Charles Sherrington in 1930? So let me go back to the PPT. Yeah. So Lars Lexell, he was very interested in trying to follow up what. So Eccles and as Eccles was also a student of Charles Sherrington, right? He had discussed this in the previous classes that Jack Eccles was a student was the last student of Charles Sherrington, and both of them were working on spinal reflexes. So spinal cord reflex action they were trying to understand. So in that case, in around 1930s or so, 1920s, 1930s, Eccles and Sherrington had made one observation. And the observation was that there were certain thin fibers in the motor nerves which which were not, although like if you stimulate these fibers, they are motor neuron fibers, right? These motor neuron fibers were synapsing onto the muscles, but when these fibers were stimulated, they did not cause any detectable contraction of the muscle. But the function of nerves is to stimulate muscle contraction. But these were not doing that. So, this was the observation that Eccles and Sherrington had made. Now, Lars Lexell wanted to follow that up. So Lars Lexell was in Sweden, right? So he was at the Karolinska Hospital in at Stockholm in Sweden, and Sweden was relatively not as affected by the world war that was happening at that time as compared to the other countries. So Lexell was carrying out, he was a young neurosurgeon at Karolinska Hospital, and he was interested in trying to find out what is the function of these thin fibers. So he realized that these fibers are actually the ones that innervate the muscle spindles. So in this diagram, if you can see, this is the this is the muscle itself. In the muscle, there are extrafusal muscle fibers, so the ones that you can see on this outside. And then there is a capsule-like structure. There's this capsule-like structure which is encased by this sheath. And in this capsule, there are muscle spindles. And these muscle spindles we have also come across in some of the previous experiments that were discussed, that these muscle spindles, their work is not to contract. These extrafusal muscle fibers, their work is to contract and carry out the muscle contraction as normally. But these intrafusal muscle fibers or the muscle spindles that are encased in this long fusiform capsule, their function is to actually uh, identify the speed and the velocity of the stretch of the muscle. So, how much is the muscle stretching, and at what speed is this muscle stretching? That is the purpose of these muscle fibers. So these muscle fibers have these muscle spindles have these 1A and 2 um sensory afferents. So the information that the stretch information is carried away from these muscle spindles by these 1A and these 1A and 2 sensory afferents to the spinal cord. So that is the function of these muscle spindles. And then there are these gamma motor neurons. So the ones that these thin fibers, right, in the motor nerves that were not causing any detectable contraction, these were actually these gamma motor neurons. So these gamma motor neurons are also the ones that synapse with these, are the ones that synapse with these muscle spindles. The alpha motor neurons are the ones that synapse with the extrafusal muscle fibers, the ones that are responsible for muscle contraction. Whereas these gamma motor neurons, they are the thin fibers that synapse with the exclusively with the muscle spindles. So now what happens is whenever there's a, so you can see that these 1A and 2, sorry, just a minute, let me. Yeah. So these 1A and 2 sensory neurons, they are basically carrying away the information from the muscle spindle, and they are present at the center of the muscle spindle. So they are at the center of the spindle, and they have this flower spray ending. So they basically have these branches, and from there, they carry the stretch information away from the muscle spindle to the spinal cord. And these gamma motor neurons, they are present at the ends, at the polar ends. So they are not present at the center, they are present at the ends. So Lars Lexell found that these thin fibers that are there, they are the ones that are actually innervating these muscle spindles. And what happens is so there is basically a regulation of the stretch of the muscle fiber that is getting integrated with the muscle contraction. So basically these gamma motor neurons are telling the muscle spindle that this muscle is contracting right now. And these sensory neurons are taking the information to the spinal cord that this much muscle is getting stretched. And Lars Lexell also found that the amount of so if these neurons are fired, these gamma motor neurons are excited, right? Then there is also excitation in these sensory neurons. So whenever there is excitation of the gamma motor neurons, there is also impulses that are detected in the sensory neurons. So that way, the spinal cord gets to know that whenever there is a muscle contraction, how much stretch is happening in that particular muscle. So the both information are being delivered, that there is also muscle contraction at that particular point in time, and also there is this much amount of stretch and this much speed of stretch that is happening. So Lexell was the one who found out about this. So that's why they think the signaling of the spinal cord, the signaling to the spinal cord regarding the state of shortening and lengthening of the muscles, as well as the information about the muscle contraction. And why this is important is because there are certain cases, like for example, in spasms, what happens is that there is excessive stretching and repetitive muscle contraction that happens, right? So there are disease conditions in which the relevance of this is very important. So coming back to the question, previous year assignment question, what was the problem that Lexell addressed? The problem was that there was a lack of detectable muscle contraction within electrical stimulation. I hope this answer is clear to everyone. The first option is incorrect because there is electrical stimulation in the motor nerves. It is not a lack of electrical stimulation in the motor nerves. There is electrical stimulation in the motor nerves. Only that Eccles and Sherrington saw that the muscle is not contracting in response to the electrical stimulation. And the large population of thin fibers was the motor nerves, not the sensory nerves. The gamma motor neurons were the large population of thin fibers. Okay, next. Uh, what synaptic pathways were identified by Jack Eccles in the early 1953? So again, let me go back to the text. It will be much easier for you to understand. Yeah. So, like we know, Eccles was the last student of Charles Sherrington, who was a pioneer of the studies in the spinal cord, especially the reflex action. Now, many of the things had been identified by Eccles and other students of Charles Sherrington and other collaborators and so on. But there was still one thing that was left. That was the circuits that were no, the complete circuit was still there was some of the things that were left to be identified. So what was that? So in previous week lecture videos, you have seen that Eccles and his collaborators in when they were in Australia, they were dissecting the pathways that were involved, the nature of the synapse, and so on, right? So Eccles summarized all of this information in his book in 1953, that is the neurophysiological basis of mind. Then, while he was investigating the cat spinal motor neuron, he discovered that there is one that is the monosynaptic pathway, in which this is a monosynaptic excitatory pathway. So this monosynaptic excitatory pathway is from the 1A muscle spindle afferents motor neurons. So what does that mean? So when there is a stimulus, for example, we'll talk. I'm explaining the tendon tap uh, reflex action, which most of you would have come across at some point during your high school, right? So that there's a tendon. So when the tendon is tapped lightly, there is a stimulus. Now this causes a stretch of the quadriceps muscle. So, I mean, as in the quadriceps muscle, this is interpreted as a stretch of the quadriceps muscle. Now, when that happens, the sensory neurons from the quadriceps muscles, the sensory afferents, basically the 1A, basically from in the muscle spindle that is present. So we discussed this muscle spindle, right? So in the muscle spindle that is present in this quadriceps muscle, there are 1A sensory neurons. So these 1A sensory neurons go and synapse directly with motor neurons in the spinal cord. Okay, and now these motor neurons, they synapse back to the quadriceps muscles. So there is a monosynaptic excitatory pathway from the 1A sensory neurons in the muscle spindle of the quadriceps muscle to the motor neurons in the spinal cord. So there is a synapse here, right? The monosynaptic means that there is a single synapse. So this 1A sensory neurons is synapsing only with these motor neurons. And now when it synapses, so it's an excitatory pathway, right? So what that means is that there is excitation of this motor neuron. Now this motor neuron, when it gets excited, it goes back and it excites the quadriceps muscle. And that causes the quadriceps muscle to contract. The muscle, because motor neuron will go, and if the motor neuron goes and activates the muscle, it is going to contract. Now, because of the contraction of the quadriceps muscle, the leg stretches, right? That is how the reflex action is working. So he observed this monosynaptic pathway that is happening, that because of the this particular synaptic circuit. So this was one of the circuits that one of the pathways, synaptic pathways that he discovered. Okay. The other synaptic pathways that he discovered were One was a so at that time it was thought that it's a disynaptic pathway. Now we know it's an oligosynaptic pathway. So he discovered a disynaptic inhibitory pathway from 1B muscle spindle afferents to antagonistic muscles. Now, what does that mean? So now, if the when the tendon is tapped, this quadricep muscle, we discussed that the 1A afferents from the muscle spindle, basically the sensory neurons, are going to the spinal cord. And then they are going to synapse with the motor neuron for the constriction of the quadriceps muscle. But there is also this other muscle that is an antagonist, right? We know that there is an agonist, there's an antagonist. There are always two sets of muscles that are present. So the antagonist of the quadriceps is the hamstring. So the one over here, this is the hamstring. It is the antagonist of the quadriceps. Now, when the quadriceps is getting, at the time the quadriceps has to contract, the hamstrings should be relaxed, right? That means the hamstring should not also contract at the same time. It should be relaxed. That is if both of them contract, that will be a problem. So when the quadriceps is contracting, the hamstring has to relax. But how will the hamstring know that it has to relax? So for that, when the sensory afferent is going to the when the information is going from the muscle spindle, when the sensory neurons are carrying this information to the spinal cord, the 1B afferents, basically the 1B sensory neurons, what they do is they synapse with certain interneurons that are present in the spinal cord. These when they so one thing that you have to know is that these sensory afferents are always excitatory in nature. They are not inhibitory. It has been shown that they're excitatory in nature. So when they synapse with these interneurons, they excite this interneuron. And now these interneurons that are there, it may be one or several interneurons. So now these interneurons that are there, they in turn will release, for example, an inhibitory neurotransmitter like GABA. They will inhibit the motor neurons that synapse with the antagonist muscle, in this case the hamstring. So these interneurons will inhibit the alpha motor neurons of the flexor. So we know that alpha motor neurons are the ones that synapse with the muscle, the extrafusal muscle fibers, right? We discussed this. So the alpha motor neurons synapse with the extrafusal muscle fibers that are responsible for muscle contraction. So what these interneurons will do is they will inhibit the alpha motor neurons of the hamstring. And because they will inhibit the alpha motor neurons of the hamstring, that means that the hamstring will not contract. The hamstring will be in a relaxed state. That will allow easier. It will make it easier. So the hamstring will be relaxed, and the quadriceps will contract, and the leg will move. This is something that we now know as reciprocal inhibition. So this is also one of the synaptic pathways that Eccles discovered. What was the third synaptic pathway that Eccles discovered? The third pathway was a disynaptic pathway from Golgi tendon organs. So the one that we just discussed, the one from the the one from the 1B sensory afferents to the antagonistic muscles, that is actually an oligosynaptic pathway. So what does that mean is that there might be several synaptic connections. It is not just one or two synaptic connections. There are several. They might, so for example, the 1B might synapse with another interneuron, that is the 1A interneuron. And then that goes and inhibits the alpha motor neuron of the hamstring, for example. So that one was an oligosynaptic pathway. The third one is a disynaptic pathway from the Golgi tendon organs. So now what is that? That is basically, so we discussed that the antagonistic muscle will relax and that will make it easier for the leg to contract. Okay. But the agonist muscle, the agonist muscle that is contracting in the first place should also be controlled. There should not be too much contraction also. Everything should always happen in moderation. Everything should always have a control on it. There should always be some kind of feedback. Biological systems always work on some feedback mechanism. And that is very apparent over here because these Golgi tendon organs that are so basically that are close to the tendon that has been stimulated, these Golgi tendon organs will know when the agonist muscle is is getting stretched. So when that happens, these Golgi tendon, so they will respond basically to the tension. So this is something called proprioception. These Golgi tendon organs will respond to the tension of the tendon. So when there is stimulation of this tendon by the tapping of the tendon, these Golgi tendon organs will give the information again from 1B afferent to the spinal cord. And that will cause the inhibition. So these, when they go to the spinal cord, they will synapse with the 1B interneurons. And when they synapse with the 1B interneurons, the 1B interneuron will synapse with the alpha motor neurons of the agonist itself. For example, they will synapse with the quadriceps itself, which is contracting. So that will act as a feedback. So the agonist is supposed to contract and the agonist will contract, but this synaptic pathway will tell the agonist that there is a limit to which it is supposed to contract. It is not supposed to contract more than this. Now it has to stop contracting. So that feedback information is being provided by this third this other pathway, which is a disynaptic pathway. Disynaptic means there are two synapses. And which are the two synapses? The first synapse is between this 1B afferent with the 1B interneuron. And so basically this synapse. This is an excitatory synapse because we discussed, right? Thus, the sensory afferents from the sensory afferents are always excitatory in nature. So this 1B afferent is going to be an is going to form an excitatory synapse with the 1B interneuron. And this 1B interneuron is going to form an inhibitory synapse with the alpha motor neurons of the agonist. That is in this case, the quadriceps. So there are three pathways that Eccles described. One is the monosynaptic pathway, which will tell the quadriceps that it is supposed to contract. So the information of the stretch information that is sensed by the muscle spindles of the quadriceps will go to the spinal cord and that will activate the alpha motor neurons that synapse with the quadriceps itself, and that will tell the quadriceps to contract. The second is the oligosynaptic pathway, in which the information from the information goes to the 1B interneurons. So there are several synapses that are being formed over here. So information goes to the 1B interneurons, and the 1B interneurons are basically the 1B interneurons are going to synapse with the 1A interneurons, and then the 1A interneurons are going to synapse with the alpha motor neurons of the antagonist, the flexor muscle, that is the hamstring over here, and that will tell the antagonist to relax. And the third one is the feedback from the Golgi tendon organ to the 1B interneurons, which will then tell the alpha motor neurons of the agonist to stop contracting after a point. So now coming back to the question, I hope this concept is clear. Coming back to the question, what synaptic pathways were identified by Eccles in early 1953? So can any of you uh, volunteer to answer this question? Like explain everyone why you think the this answer is correct? Does anyone want to volunteer? No? Okay, in that case, let me explain it myself. Okay. Option 1, monosynaptic inhibitory pathway from warning muscle spindle afferents. We know that the monosynaptic pathway is excitatory in nature. It has to be excitatory because the monosynaptic pathway is responsible for the contraction of the quadriceps muscle. When the quadriceps muscle is getting stretched, it has to know that now it is supposed to contract in response to this stretch. So this monosynaptic pathway is excitatory. These sensory neurons, like I said, are excitatory neurons, and they will excite the motor neurons, which will go and give information to the muscles for muscle contraction. So that's why the first option is wrong. The monosynaptic pathway is supposed to be excitatory and not inhibitory. Second, disynaptic excitatory pathway from one-way muscle spindle afferents. The pathway from the one-way muscle spindle afferents is supposed to be inhibitory. We just discussed that it is not supposed to be excitatory, it's supposed to be inhibitory. Third, monosynaptic excitatory pathway from Golgi tendon organs. No, the pathway from the Golgi tendon organs is, first of all, it is disynaptic, like we just discussed, and second, it is supposed to be inhibitory. It is supposed to give a feedback for the muscle contraction to stop. So it is a disynaptic inhibitory pathway. That is why the third option is also not correct. And the final option, disynaptic inhibitory pathway from 1A muscle spindle afferents. Yes, because the 1A muscle spindle afferents are giving a disynaptic inhibitory, although now we know it's oligosynaptic, but he identified it as a disynaptic inhibitory pathway from the 1A muscle spindle afferents to the antagonist muscles, right? This one. So this antagonist muscle is supposed to relax. So this pathway is supposed to be inhibitory. These interneurons will inhibit the alpha motor neurons of the flexor. So I hope the answer is clear to you that the synaptic, what were the synaptic pathways that were identified by Jack Eccles. Next, in the 1950s, which pathway was referred to as the Renshaw cell pathway by Eccles? So, what was observed was that in the spinal cord, when they were trying to excite the axons of or in the ventral root axons, they observed that the information in the axons, when they were they were stimulating the axons, the information in the axons was going antidromically. Antidromically means in the opposite direction to the one in which it's generally information travels. That means the information was traveling from the axon to the cell body. That was one thing that they were observing. And second thing that they were observing was that there was hyperpolarization of the cell body. So these were the two observations that they made. One is that when the axons were getting stimulated, there was this is not okay. Yeah, there was antidromic action potential, which means that information was going from the axons to the cell body, followed by a long-lasting hyperpolarization. This was something they observed by intracellular recording. So they did not understand why there was this hyperpolarization because they were only stimulating the axon. They discovered that there were certain interneurons that were getting activated by collaterals of the axons. So what does that mean? That means that so uh, if the branches of axons are called collaterals, axon collaterals. So here you can see that there is this collateral branch coming from this axon, right? So these axon collaterals were synapsing to, so these I mean, the axon was getting excited. So this axon was giving an excitatory output. But these collaterals were synapsing with certain interneurons. And these interneurons were taking this information of the information that this axon was excited, they were receiving this information from the synapse. And having received the information, they were synapsing with the cell body of the same neuron and they were inhibiting it. So now here the synapse that was formed was inhibitory in nature. These axon collaterals were forming excitatory synapses, whereas these interneurons, which were receiving information from the axon collaterals, were synapsing back to the same neuron and they were inhibiting the neuron. Now, this kind of phenomenon that they observed was they called it as the Renshaw cell pathway. And why did they call it as Renshaw cell pathway? That is because there was a young investigator called Birdsey Renshaw. He was working at Harvard, and he had made some recordings, and from those recordings, he predicted that there might be some interneurons that might exist that are actually doing this kind of they were that are actually carrying like they're causing this kind of phenomenon, that they were being activated by the collaterals, but they were synapsing onto the same neuron and they were causing hyperpolarization similar to those that are seen in case of motor neuron IPSPs. So IPSPs are inhibitory postsynaptic potentials. But Birdsey Renshaw died very young. He died in 1947 of polio. So, but because Renshaw was the first one who had made such a prediction, so Jack Eccles decided that he will name these interneurons as Renshaw cells in memory or in to commemorate Renshaw's contribution, like his idea that there might be these kind of cells. So he named them as Renshaw cells, the inhibitory interneurons that are carrying out such a function. So in the 1950s, which pathway was referred to as the Renshaw cell pathway by Eccles? So the correct answer is recurrent inhibition pathway. Why recurrent inhibition pathway? Because whenever the muscles are, axons are getting stimulated, there is a recurrent inhibition, or it is happening again and again. So there is a recurrent inhibition of the motor neuron by the axon collaterals through the interneurons. So this pathway recurrent inhibition pathway was referred to as the Renshaw cell pathway by Eccles. Next, who first described and explained the phenomenon of contrast enhancement in the late 19th century? So let us understand what does that mean. So for example, if you take a look at this picture, you will see that wherever there are these borders between the different bands, you will see this. I mean, visually when you visually there it will look as if there is a lighter band over here and there is a darker band over here, right? So whenever there is a lighter band, I mean whenever there's a lighter band that is close to a darker band at the border or at the very edge, you will see that it feels that the lighter band looks more lighter and the darker band looks more darker at the border. If you look carefully, you will observe that this is true for you. At each of these edges, it will look, you will perceive it like that. That all, I mean, although this band is uniform and this band is uniform in terms of the color, but at the border, if you see, it will look as if the lighter band is looking lighter and the darker band is looking darker. So this is the phenomenon of contrast enhancement. And this was first described and explained by Ernst Mach. Although, like, he did not explain the reason for this kind of phenomenon, but he explained that this kind of phenomenon does exist. And these bands are therefore called as Mach bands because he was the one who first described and explained this phenomenon. So, in the text also, if you see, yeah. So if you see here also, it is very like whenever you see these kind of different color bands that are uh, all close together, you will see these kind of, the lighter band is looking lighter at the edge and the darker band looks darker, even darker at the edge. Now, the reason for that actually is because on the light side, there is, so in terms of the neural in the way the visual circuit was working. So what happens is in the light side, there is less inhibition from the dark side. So there's a phenomenon called lateral inhibition, which slowly these people started understanding that it exists, and now we understand that because of the lateral inhibition of like there is a phenomenon called lateral inhibition in which neurons can like when they are activated, neurons can inhibit the neighboring neurons. So basically, that is the concept of lateral inhibition. So what happens basically is that on the light side, there is less inhibition from the dark side, and that makes it lighter. On the dark side, there is more inhibition from the light side, making it dark. So that is actually what is happening at the light dark border. And because Ernst Mach explained this phenomenon of contrast enhancement in 1897 for the first time, these are called as Mach bands. Next question. Who initiated experiments on crayfish in the 1930s and reported that a single shock to a giant axon elicits a tail flip in crayfish? So, again, let me go back to the text. Yeah. So, Seiji Wiersma or Kees Wiersma was the one who tried to use the crayfish to understand reflex behavior. So he used the crayfish because the crayfish has several giant axons, and these giant axons innervate the ganglion cells to the muscles of the body. And it was much easier to visualize these, I mean, as in study these axons and understand that, like, because of the less number of, less number of it less complexity of the organism, it was much easier to study them. And because of the axons were also very large, so it was much easier to stimulate the axons and study the responses. Always remember that it is much more like we have, like history has always shown us time and again that an experiment that is simple but elegant in terms of trying to address the question is way better than an experiment that is just complicated for the sake of it by using very high end equipment. Because an elegant experiment using very simple apparatus, as long as they are able to address the question sufficiently, is something that will give us much more information than a more complex system in which there are you and complex equipment and trying to discern something from that. And uh, that is how these days also invertebrate models, for example, C. elegans and the fruit fly, Drosophila melanogaster, and so on, they are they allow us basically, and even for example the limitless, the crab that was used by several people, and the squid axon, and the several invertebrate systems that were used, those those gave us, and those continue to give us a lot of information about the nervous system. And because of their relative simplicity, the ease of manipulating them, the ease of carrying out genetic manipulations, even at the single cell, at the single the identification of different and different types of cells or different components and so on, it makes it much easier for us to use those and understand what is happening. And because many of the principles of neuroscience are do happen to be conserved, many of them do happen to be universal. So understanding neuroscience using those models makes it much easier to extrapolate several of the principles that are discovered in those organisms. And this again is one more example of that. So, what did Wiersma do? Wiersma was studying the reflex behavior in crayfish. And crayfish has several giant axons, and these axons are innervating the cells that synapse to different muscles of the body. So, in 1947, when he gave a single shock to a certain axon, he saw that there is a tail flip. So the crayfish has a tail, which he saw that it's suddenly flipping when he is giving a single shock to a single axon. So this indicates a very complex behavior, right? Because the crayfish generally flip their tail as an escape response. So it's a very coordinated escape, a behavioral response at a very large scale that is being carried out by the stimulation of shock of a single axon. So that indicated to him that uh, the crayfish, so he there was one thing that was like that one thing that came out of this was that even the crayfish nervous system, an invertebrate nervous system, has reflex similar to that of the vertebrate spinal cord. So even invertebrates have nervous system that have uh, reflex actions and hardwired responses and so on. which are similar to those that are seen in the case of the spinal reflexes that are present in the vertebrate systems. So from that, Kiasma came up with this idea that there might be certain command neurons. So basically, there might be certain neurons which can command the whole system, the to carry out a single complex behavior. For example, here, just the shock to a single axon is giving rise to such a complicated, complex response. A large scale behavioral response. So he came up with the idea that there might be certain command neurons which if those neurons are activated, and then those will command the circuits to give rise to a single behavioral output. So, Piasma also came up with the idea of central pattern generators. So, what those are? They are central pattern generators are basically they carry out rhythmic behaviors, rhythmic reflex actions. For example, the locomotion, peristalsis, and so on. So he saw that there are these central pattern. He and several others saw that there are these command neurons and there are central pattern generators and so on. So coming back to the question, who initiated experiments on crayfish and showed that a single shock to a giant axon elicits a tail flip in crayfish? It was C.H. Wiersma. And what is the significance of that? The significance is that there are such uh, so the tail flip, like I said, is a behavior that that is for the escape response, right? So the crayfish carries out such a behavior for escaping. So such kind of circuits that are present, such kind of pathways that are present. Sorry, let me just. Okay, Soumya, I'll explain the central pattern generators. Let me just finish telling you about the significance of this. So, such escape responses and such, so these kind of things are evolutionarily important, right? Because it implies that there are certain circuits, such certain hardwired circuits that are present, which are required for the survival of the organism, and those have come across. Those have come into place during the course of evolution to help the organism to survive. So, and the fact that, that is the reason that such kind of command neurons and such kind of CPGs exist in the first place. Okay, regarding central pattern generators, so this is something that is still being studied even now. People are trying to understand what central pattern generators are, how they work, and so on. So, what central pattern generators are basically those circuits or those pathways that are responsible for rhythmic actions. So they consist of both generator elements and driver elements. But they are later people understood that these drivers also consist of even more generators and modulators and so on. So this is something that is still being studied. It is going into the depth of CPGs would be beyond the scope of this particular discussion. But there's a controversy or there is a debate that CPGs may not exist in vertebrate systems, they may not be present in humans. So, this is actually still a region of a lot of ongoing studies. And CPGs, for example, are responsible for the rhythmic rhythmic locomotion activities of worms and so on. So, anything that involves any rhythmic activity or any circuit that is similar kind of like a reflex behavior, you can and those can those do come under the purview of CPGs. But this is still something that is a part, like that is for which studies are, I mean, there's issues and studies are still ongoing regarding CPGs. So I'll restrict the discussion to that, because discussing CPGs in greater detail would be deviating from the main topic. But if you would like to discuss this with me in further detail, I would be happy to discuss this maybe in the discussion forum, or maybe you can email me and we can discuss this. Next, who was one of the first to insert a microelectrode into the cortex of the anesthetized cat to record different types of cell activity in different layers? So, this was done by Herbert Jasper. So, oh, by the way, I would strongly recommend you all to go through these papers. These are some of the very seminal papers that describe many of the experiments that we are discussing right now. For example, the one by Eccles, in which he discovers the in which he describes the Renshaw. I mean, first. Uh, in the first paper, in which he talks about the inhibitory synapses, basically the recurrent inhibition pathway of the Renshaw cell pathway. So I would really recommend you to go back and check these papers out. At least try to look at what these authors have, what are the brilliant ways in which these authors have tried to understand these phenomena. Because remember, at that time they did not have access to those resources that we do have now, but in spite of that they have managed to outline whole pathways for us to like understand, right? The whole, the pathways that we, the synaptic pathways, the spinal reflex pathways that we discovered, for example. Just imagine, all they had were a very primitive equipment, but they managed to actually find out these whole synaptic pathways, which according to me is remarkable. It's brilliant that they were able to actually understand these pathways just using those kind of primitive equipment that they had at that time. I mean, primitive in terms of, I mean, in terms of current times, those are relatively primitive, right? But still, they managed to uncover so much information. Okay, let me. Where was I? Yeah, Herbert Jasper, right? Okay, I'm not able to find it right now in the text, but yeah, so it was Herbert Jasper who actually was the first to insert a microelectrode into the cortex of the anesthetized cat, and he actually observed the different. So there are different layers of the cortex. The cortex is a multi-layered region, and he was the one who actually inserted a microelectrode into the cortex, and he tried to record the activity of cells in each of the layers. He was one of the first to do it, but then others followed soon. For example, Hubel, for example, and then even Denise was one of the people. Denise was one of the people who tried to do insert electrodes and do recordings. Richard Young was a German who was the first to insert electrodes into the cortex and carry out recordings and so on. But the first to insert a microelectrode into the cortex of an anesthetized cat and record the different types of activities in different layers of the cortex, that was Herbert Jasper. Next, who, on the suggestion by Fortes, designed wire electrodes for single unit recordings in the cat cortex and developed a tungsten electrode that was better than the previous steel ones. So I just told you about David Hubel, right? So David Hubel was actually, like many of the people at that time, he was actually someone who had started with his studies by training in medicine. So at that time, there was no. I mean, initially at that time, people who generally started training in medicine or in maybe in some other fields, and then they transitioned to studying about neurophysiology and neuroscience and so on. It was not like a direct. People did not directly start. I mean, not many people directly started studying neuroscience, neurophysiology, or neuroanatomy, and so on. Many people started their training first in medicine, and then they developed interest in research, and then they started then they devoted a lot of their efforts to research. That is how it generally happened. And David Hubel was, I suppose, it's not in this. I think it's okay. It's not a part of this, but okay. So David Hubel was one of the people who, again, who started his studies in medicine, and then he developed an interest in understanding neurophysiology. And he was at the time. Remember, war was happening. So David Hubel was actually from Canada, but he had come to the United States for his studies and so on. So, but he had a dual citizenship, so for both the United States and Canada. But that meant that Hubel had to get drafted for the war, because at the time the Second World War was happening, and whoever was an American citizen, they had to get drafted for the war. There was no other choice. They had to get drafted for the war. They had to take part in the war efforts in some way or the other. So in that case, Hubel was also assigned. He was assigned to an institute in Washington D.C. And there, it was an Army Research Institute in Washington D.C. And there, Hubel was. I mean, he could devote most of his energy to research at that place, because it was a research institute. So there he was, while he was there, he started working on these single unit recordings. He initially had no prior experience in single unit recordings. He had no experience in working on the cat cortex whatsoever. But he was told that, hey, why don't you try designing electrodes? So he designed these wire electrodes, because you need wire electrodes that can the to put inside the cell, to insert in the cell in the cortex, right? So he designed these wire electrodes for single unit recordings in the cat cortex, and he managed. Despite doing this for the first time, he managed to develop a tungsten electrode that was way better than the previous metal ones. And he used his those electrodes, and he even came up with some other interesting things. So I'll tell you about that. So the answer to this question is David Hubel, because he was the one who first managed to develop a tungsten electrode that was way better than the previous metal ones. In addition, he also came up with electrodes that could leave a trace of where they were recording. So they could deposit some material on in that region wherever the they like to whichever region they were inserted, they could deposit some when they could make some depositions. Those electrodes could deposit some material. And when the tissue, when the tissue was opened up, and you could see that, oh, this is the region where the electrode was recording. So he came up with these really good tools, and using his, I don't know, sheer creativity, I could say, and he managed to uh, carry out single unit electro recordings in the cat cortex. And what I mean by single unit recordings is that, remember, we are dealing with the cortex, basically the a part of the brain. So brain and the cortex is a region that is going to have lots of neurons very densely packed together. And in fact, we know the interneurons are very small. So you do not know whether you are recording from a single neuron. I mean, as in you're recording from the axon, whether you're recording from the cell body, whether you're recording from some other region, whether you're actually recording from, like, maybe some extra information is coming from other cells. I mean, not sure what you're recording. So you cannot say that I'm recording from a single neuron. So rather, uh, the designated term was single unit. So wherever they were, like, they managed to record to like single regions, or single fibers and so on, they used to call those as single unit recordings rather than a single neuron recordings, because it was like, a lot of cases there were errors. You did not know that this is what whatever you're recording, whether you're actually recording what you think you're recording. So, on suggestion by Fortes, David Hubel designed these tungsten electrodes, which were way better than the steel ones. Next, what distinction was emphasized regarding the motor systems, particularly in primates? Okay, so the distinction between pyramidal and extrapyramidal systems was emphasized. So what do we mean by pyramidal and extrapyramidal systems is, let me just go back to the text. Yeah. So two different motor systems were distinguished. What were these two different motor systems that were distinguished? Those were the pyramidal motor system and the extrapyramidal motor system. So the two different motor systems in the cortex were shown to exist. One, the pyramidal system, in which the cortical pyramidal cells send their axons through the pyramidal tract to make direct synapses on the motor neurons in the spinal cord. So the pyramidal cells that are present in the cortex, they send their axons through the pyramidal tract. And these axons are directly synapsing with the motor neurons that are present in the spinal cord. And these are responsible for the control of fine movement of the digits in the primates. This is one kind of motor system. So here there is a direct synapse that is being formed between the cells of the cortex and the motor neurons of the spinal cord. The axons of the cells of the cortex and the motor neurons of the spinal cord. Whereas in the extrapyramidal motor systems, what is happening is the cortical connections that are being made, those are being made to the brain stem centers. So brain stem is the medulla, that is that what we call as medulla oblongata. So the cortical connections are being made to the brain stem, and these brain stem centers are then connecting with the interneurons in the spinal cord. Then those interneurons are controlling the motor neurons. So the connections are not being made directly in the case of the extrapyramidal systems. So first, the cortical connections are being made. So the information in the cortical cells are synapsing with the those in the brain stem. Then the brain stem centers are connecting with the interneurons in the spinal cord. Then the interneurons in the spinal cord are controlling the motor neurons. And the significance of this is that it could be understood that if there is any damage, if there is any lesion, is that damage, is that has that damage happened in the spinal cord, has that happened in the midbrain, in the white matter, or the cerebral cortex? Basically, if there is a problem, if there is someone who has had a damage to their nervous system, where has the damage happened, at what level has the damage happened? Is that because we see that in the pyramidal system the connection is direct. In the extra pyramidal system there are various different connections that are happening. So that helped to distinguish the fact that there are two different systems and this is how they are working. This information helped the help people to distinguish the motor defects that are arising, the cause for the motor defects that are arising. So, what distinction was emphasized regarding the motor systems, particularly in primates? The distinction between the pyramidal and the extrapyramidal systems. Okay. What did Penfield systematically map in the human cortex and what became widely known through his findings? So, Wilder Penfield was someone who worked on several things. He, in fact, carried out various operative procedures. Yeah. So he carried out various procedures for removal of tissues from the cortex for relief of epilepsy. So epilepsy is a neurological disorder in which people have unexplained, or like people have seizures that can be that may arise due to different lesions in different parts of the brain, or even without any lesion there might be certain problems that because of imbalance of excitation versus inhibition and so on, and those can be like, and that can happen like throughout the brain, or that can be because of certain focal seizures, that is basically certain regions of the brain might be affected. So in case of where certain regions of the brain might be affected. So, Wilder Penfield at that time was trying to carry out operations to remove cortex tissue so that in some cases it might help to relieve the person of epilepsy. So during the course of that time, what he did was he also systematically mapped the motor cortex. And when he was doing that, he found the presence of motor strips in the frontal lobe. And similarly, other people also found that, for example, in the somatosensory system also it was found and so on. In basically what he found was there are certain regions that are like there's a whole map of regions that are present in the human cortex, that this particular region has cells that or has motor pathways and so on. Sorry, that has pathways and so on that correspond to the activation or correspond to the muscles of this organ, or this region in the brain has neurons and so on that will control the muscles that are present in this organ and so on. So basically, this is not just true for the motor system, but also for other systems also. Basically, there were different regions that were there that mapped to different regions of the body. So basically, this was called the homunculus or the little man. Basically, the whole body is mapped to different regions that are present in like the motor strip, for example, or in case of the somatosensory system, the somatosensory homunculus. So he showed that there are these different regions that are responsible for the whatever that is in the muscles for governing the muscles that are present in different regions of the body. So Penfield systematically mapped the motor areas in the frontal lobe to the activation of different muscles in different parts of the body. And he came up with the concept of the motor homunculus. And like I said, it was shown by other people that this is true for like other systems also. It is not just true for the motor system, it is also true for different systems, such as the somatosensory system. Okay, next. What did Adrian's experiments suggest regarding the olfactory system? So Edgar Adrian was somebody you must have come across in the previous week's lecture videos also. He is a recurring he's someone who has carried out many different kinds of experiments in many different contexts. So Edgar Adrian did experiments with regard to the olfactory system. So and what he saw was that is that there in the text? Hmm. Yeah. So what did Edgar Adrian see? He saw when he did recordings from the olfactory bulb that if you give different odors, if you give, for example, acetone, or if you give xylol, if you give pyridine, if you give odor, eucalyptus, and so on, if you give different odors, he was able to see different kinds of uh, uh, impulses that were being generated in the olfactory bulb. So, depending upon which um odor was given, different regions of the olfactory bulb were getting activated. And he proposed that there is a spatial patterning and there is a temporal patterning of the uh, odor. So the different regions of the olfactory bulb basically respond differently to different odors. So there are certain regions of the olfactory bulb, certain critical regions of olfactory bulb, which will respond, say, more strongly to acetone. Then there are certain regions that might more strongly respond to xylol and pyridine and so on. Well, like basically, he was trying to say that different odors are like activate different regions of the olfactory bulb in a different way. So there's different patterns in which each odor is activating the olfactory bulb, and that might be how the odor is getting recognized. So his idea that this kind of phenomenon is happening was called the pattern theory of odor representation. So he called this the pattern theory of odor representation, and his experiment suggested that there is a systematic relation between the olfactory epithelium and the olfactory bulb. Basically, he showed that uh, depending upon the odor, the impulses that are generated and how the system responds is different. Next, what function does the muscle spindle serve in the body? This is something we have discussed during the beginning of the session, right? For those who had not joined during that time, let me briefly give you a recap about what we discussed. So we discussed about the presence of these muscle spindles. So what are these muscle spindles? These are basically fusiform. Sorry, these are basically modified muscle fibers that are present within long fusiform capsules within the body. So basically, you can see that there is this normal muscle fibers that are responsible for contraction, which are called the extrafusal muscle fibers. Then there is a fusiform capsule, which has a sheath to it, and inside that are intrafusal muscle fibers or the muscle spindles, which are basically modified muscle fibers. And the function of these muscle spindles is to basically inform the spinal cord about the extent of stretch of the muscle. How much is the muscle getting stretched? So that is the information. So whenever there is a stretch of the muscle, the muscle spindles are the information will be received by these muscle spindles. So because of the contraction of the muscles, the stretch information and the speed and velocity of stretch that is happening will be carried away from these muscle spindles by the sensory afferents. Afferents means those that are going towards the spinal cord or away from the organs or away from the muscles towards the spinal cords. So afferent is towards the spinal cord, efferent is away from the spinal cord. So these are afferents. The 1A and the 2 are the sensory afferents, or they are the sensory neurons that carry the stretch information away from the muscle spindle to the spinal cord. And the alpha and the gamma motor neurons are the efferents. So the alpha motor neuron will synapse with the extrafusal muscle spindle, sorry, the extrafusal muscle fiber, and the gamma motor neurons will synapse with the intrafusal muscle spindles. So the alpha and the gamma motor neurons are efferent neurons because they are going away from the spinal cord to the muscles or the sense organs. So now keeping this in mind, what function does the muscle spindle serve in the human body? The muscle spindle, it does not generate nerve impulses for movement. That is not the work of the muscle spindle. That is that is something that requires a lot of other information. Integration of, that is a very higher order phenomenon. Signal the spinal cord about muscle stretching and contraction. We just discussed that. That is correct. That's what the muscle spindles do. Produce neurotransmitters like acetylcholine. No. Muscle spindles are basically modified muscle fibers. Why will they produce neurotransmitters like acetylcholine? That is the function of motor neurons. Control the heart rate through motor neurons. No. They control they give information about the muscle stretching and contraction. Their function is not to control the heart rate. Next, what did Renshaw cells contribute to the understanding of spinal cord pathways? Okay, this is also something we discussed a little while ago, but let me just briefly explain it again, very, very briefly. Yeah, so we saw about the Renshaw interneurons, right? What did we see? We saw that So this is something that was done by Jack Eccles, remember? Jack Eccles and his collaborators, when they were studying when they were exciting the axons in the spinal cord, in on the ventral side, they saw that when they were stimulating those axons, they saw that the motor axon collaterals They saw that the motor axon collaterals are synapsing with certain interneurons. So motor axons have collateral branches, which are excitatory in nature, right? I mean, the motor neurons, when they get excited, the collaterals will also give excitatory output, correct? So these excitatory output that is received by these interneurons, but these interneurons then go and inhibit the motor neuron itself. So these interneurons form synapse with the cell body, with the same motor neuron, and they inhibit it. So this is a kind of feedback inhibition that they are carrying out. And these interneurons were called as Renshaw cells. These were termed as Renshaw cells by Jack Eccles, and this pathway of recurrent inhibition was called as the Renshaw cell pathway. This is something we have discussed. So now coming back to that question, so, what did Renshaw cells contribute to the understanding of spinal cord pathways? They told us that there is a phenomenon of recurrent inhibition via interneurons and motor axon collaterals. Because these Renshaw cells that Jack Eccles and his collaborators found, this was in the spinal cord. Because Jack Eccles is someone who did who carried out most of his studies with respect to the spinal cord reflexes, and while he was doing that, he found that there are these inhibitory interneurons that can cause feedback inhibition of the motor neurons themselves. Next, what is the significance of Builder-Penfield's cortical mapping? So this is also something we just discussed. Builder-Penfield was the one who, during his mapping, during while he was mapping the frontal lobe, he saw that there are just a minute. Yeah. So while he was carrying out a systematic mapping of the human cortex, he saw that there are motor strips that are present in the frontal lobe, and from that he came up. He termed this as the homunculus or the motor homunculus. Basically, the idea that there are different regions in the motor homunculus which are responsible for individual contacting or controlling muscles of individual regions of the body. So that is the significance of Penfield's cortical mapping work. Next, which two motor systems were identified based on their functions? We know that there are two motor systems that were identified. One is the pyramidal motor system and the other one is the extrapyramidal motor system. So the pyramidal motor system is one in which there are direct synapses that are being made by the pyramidal cells in the cortex with the motor neurons in the spinal cord. And these direct connections help in controlling fine movements in the digits. Digits means basically the fingers and the toes. So to control the fine movements of the digits in primates, these pyramidal motor systems are responsible. And the other one is the extrapyramidal system, in which the connections are not direct, the connections are indirect. The connections are between the cortex with the brain stem centers, from the brainstem centers to the interneurons of the spinal cord, and finally of the interneurons with the motor neurons. So these two different systems were identified, and these were identified on the basis of their functions. Next, which cells in the retina did Keffer-Hartline study for visual signal processing? So Hartline studied the limulus polyphemus, or the crab. Wait a second. Hmm, yes. Keffer-Hartline studied the horseshoe crab, Limulus polyphemus. And he studied the visual system. And why did he use the Limulus polyphemus? Is because the visual system of this crab has uh, light sensitive receptor cells, and these light sensitive receptor cells surround an eccentric cell. So this eccentric cell, so whenever these light sensitive receptor cells get activated, they stimulate the central eccentric cell, the central dendrite or the central eccentric cell. And then the information goes from the eccentric cell to the like via the optic nerve to the visual system. I mean to the visual center. Now, uh, what is the advantage was that because this the optic nerve of the limulus could be dissected to even single fibers. So whatever whenever they stimulated the receptor cells, that resulted in the stimulation of the eccentric cells, and whenever these eccentric cells were getting stimulated, the impulses could be recorded from single eccentric cells because of the fact that they could dissect the optic nerves to single fibers. So that's why Kiefer Hartline used the Limulus visual system for his experiments. Now the question is, what cells in the retina did Kiefer Hartline study for visual signal processing? So we now know the answer is eccentric cells in the horseshoe crab retina. And there was something that uh, Keffer Hartline suggested like at that time itself, that he suggested that we should use comparative physiology. Comparative physiology means basically he suggested that you should use organisms like other organisms like invertebrates, and because the visual system is something that can be studied in various as in like all on like all, many organisms have a well developed visual system, right? So you can study the visual system using different types of organisms, and you can compare the information from that, and then you can extrapolate that to what actually happens in mammals and humans and so on. So the eccentric cells of the retina is what Keffer Hartline used, and those were from the horseshoe crab retina. Next, what feature of Renshaw cell inhibition was discovered by Eccles and colleagues? This we have discussed several times, and the feature that Eccles and colleagues discovered was that the motor neuron was getting hyperpolarized. Whenever the axon was getting stimulated, the motor neuron was getting hyperpolarized. And why was that? That was because the collaterals or the branches of the motor neuron axon, those collaterals were synapsing with the Renshaw cells. And then those Renshaw cells were, or the interneurons were inhibiting the motor neuron itself, causing the hyperpolarization that Eccles and colleagues observed. Next question. What is the role of the pyramidal motor system? The role of the pyramidal motor system is to control the fine motor, is for basically the fine motor control via direct spinal cord synapses. So whenever you need any fine control, because uh, we do not really appreciate the extent to which we use our hands for, like, say, playing the piano, playing musical instruments, holding different things, typing, so on. These are actually uh, actually movements that require very fine coordinated impulses, right? And that is something that is affected by the pyramidal motor system. So the answer to this question is fine motor control via direct spinal cord synapses. That is the role of the pyramidal motor system. Next, what did Jerry Letwin's work on the frog's retina reveal? So what did Jerry Letwin do? Does anyone know? Can anyone tell me? What did Jerry Letwin do? Very briefly, maybe one or two sentences? No? Okay, let me tell you then. Jerry Letwin. Yeah. So, we had discussed about Heartline's work on the retina. We discussed about the fact that people were trying to understand different regions of the cortex, right? So basically, because the cortex is, like, has systems that are responsible for somatosensory, like there are somatosensory centers, there are motor centers, and so on, right? So people were trying to understand also that the visual center that is there in how is the processing happening, how is the information that is being seen by the retina, how is that information being processed, what kind of processing is happening in the brain of the sensory information, and how are features being detected. So that is something people still hadn't found at that time. So Jerry Letwin, when he was working with the frog optic tectum. So tectum is basically a region that is present in the central brain of the frog. Uh, this when he was trying to work with when he was working with the frog optic tectum along with his colleagues. So he tried to understand the responses of the retina from the optic nerve endings in the optic tectum. So basically from the retina via the optic nerve to the optic tectum. This was the whole pathway, and he was trying to understand how it is that I mean the paper that they published, what the frog's eye tells the frog's brain. So how is the information, what kind of information is going from the retina to the brain, and how is it processed? So they observed that there is, like, there are ways by which the retina is detecting and the information is getting integrated, so such that they are able to detect features, they are able to detect curvatures, they are able to detect borders, and so on. So they are able, I mean, the frog retina is able to detect different kinds of features, and that is this visual stimulus is getting integrated in the optic tectum. So this is this work by Jerry Letwin and his colleagues. That is the one in which first the idea of feature detectors in the brain came up. So his work on the frog's retina revealed that the retinal ganglion cells act as feature detectors. So they can detect different shapes, they can detect different curved surfaces, and so on. Next, what technological breakthrough allowed Richard Young to advance visual cortex studies? So, what did Richard Young do? Richard Young was the first one to carry out microelectrode recordings in cats. He was the one who first inserted microelectrode recordings in the cortex of cats and tried to study it. I think he did this in 1952, if I'm not wrong. And he was the first one to attempt uh, inserting microelectrodes in the cortex. And then other people followed. We know that there was David Hubel, there was Dennis, there were several other people who later who started taking up these studies by using microelectrodes to study the visual cortex. But Richard Young was the first one to do that. These are some things that we have discovered that discussed previously. I mean, that were discussed in the previous lecture videos about the electron microscope, the Nernst equation, and so on. Right. So that those were not things that had anything to do with Young. Finally, last question. What theory of olfaction was developed by Edgar Adrian? So, what did Edgar Adrian do? Edgar Adrian, we had discussed this also. So in the early 1950s, he reported multi-unit recordings from the olfactory bulb of rabbit. And what did he show? He showed that different odors elicit different patterns of activity across the olfactory bulb. And he suggested on basis of that he used different odors. We discussed that he used acetone, he used what was that, he used eucalyptus. He used many different kinds of odors, and by using those he showed that, yeah, xylol, pyridine, and so on. By using these different types of odors, he saw that using each of them elicits a different pattern of activity. And he suggested that the odors are encoded by different spatial and temporal activity. That is how the system knows that this is the particular odor that it is sensing. And he called this the pattern theory of odor representation. So the theory of olfaction that was developed by Edgar Adrian was the pattern theory of odor representation. So with that, I come to the end of the previous year questions. Uh, in case, does anyone have any doubts that you would like me to address? If if you have any doubts, feel free to put it up in the chat box or unmute yourself and ask me, whichever you're comfortable with. Otherwise, you may also put it in the discussion forum, and I'll be happy to answer your questions in the discussion forum. If you have any particular questions regarding the live interaction session today, uh, you can you please explain the hyperpolarization of motor neuron collaterals? Okay. Let me go back to the. Yeah. See, now what happens is, okay, two things that you need to understand. Whenever there is depolarization means that there is excitation that is happening. If there is hyperpolarization, That means that there is inhibition that is happening. Okay. Generally, depolarization refers to an excitatory event, and hyperpolarization refers to an inhibitory event. Okay. Now, what Eccles and his colleagues found was that they were giving shocks. They were activating the axon. Okay, so when the axon was getting activated, or they were giving impulses to the axon, they saw that first of all the information is going antidromically. That is, it is going from. They saw that there's a impulse that is going from the axon to the cell body. That is one thing they saw. Second, they saw that there is hyperpolarization of the motor neuron. So the cell body was getting hyperpolarized. Now, hyperpolarization means that there is some inhibition. That means that there are some inhibitory currents that are coming to the cell body, right? But they had not done anything such that the cell body will get hyperpolarized, right? Because they were only stimulating the axon. They were only trying to excite the axon. But they were saying that the cell body was getting hyperpolarized when they were in fact exciting the axon. So they tried to find out the reason. And they found that these axons have collaterals, or they have branches. Okay, collateral means that there are branches of the axon. So these branches are interacting with, or they are synapsing with these interneurons, and this is obviously excitation. The axon is getting excited, the collaterals are also getting excited, and this synapse is also excitatory. So this in the input that the output that is going from the collateral to these interneurons is an excitatory output. Okay, but these interneurons, having received the information, they are synapsing with the motor neuron itself. This here, these synapses, these are inhibitory, meaning they are releasing an inhibitory neurotransmitter, for example, GABA, such that the binding of GABA to its receptors will result in hyperpolarization of the cell body, and that causes inhibition. Is that clear? Did I answer your question? So Soumya, the motor neuron collaterals themselves are not getting hyperpolarized. That is not the point. The point is that uh, the motor neuron is getting excited. The motor neuron is getting excited, but there is a feedback that is happening. The information that is going along the motor neurons, the impulse will also pass through the collaterals, right? So that information is an excitatory information. The output that is received by the interneuron is excitatory, but the interneuron is responsible for converting that to inhibition of the motor neuron itself. Basically, a feedback loop is being generated. So whenever the motor neuron is excited, in some time it also gets inhibited. I mean, it also stops getting excited basically. So basically, there is a feedback that is happening. Okay, you should get excited only for so long. You should not get excited for longer than this. So basically, the whole body always, the human body always relies on feedback systems in various contexts. For example, even in case of temperature, there is a feedback system. In case of, we saw in case of the quadriceps and the hamstring, we saw the feedback system. So there is always a feedback to keep things in check. And when there is a lack of feedback, or when there is a disruption of feedback, that is when diseases happen. So, for example, in case of epilepsy, what happens is there is an imbalance in excitation and inhibition. So, in epilepsy, there is an imbalance of excitation and inhibition, and that causes the disease to happen. That is a prevalent idea about how epilepsy happens. So, I hope that is clear. So, if no one has any more questions, I will just a minute. Yeah, these also I would urge you to read. This is the one by Penfield regarding the homunculus, the motor homunculus, and this one by Letwin is the one we discussed about what the frog's eye tells the frog's brain. Basically, the idea that retinal ganglion cells can act as future detectors. Then, yeah, the paper by in which this was of course in German by Richard Young, who was a German, in which he was, like we discussed, the first one to insert microelectrodes into the visual cortex of cat and make recordings from the visual cortex. So with that, if no one has any further questions, I'll stop the meeting. Thank you very much for attending the class. Uh, Please feel free to share your. Thank you, Amrita. So please feel free to share or if you have anything to discuss or if you have any questions or doubts, please feel free to put them up in the discussion group, discussion forum. I would be happy to answer any of your questions. I'll be sharing the class. Thank you, Shruti. Yeah, so I'll be sharing the YouTube video link.
Hello, man.
Yes, Diva.
Yeah, I've turned on the mic to thank you. So this is my first class attending on Saturdays. So
Okay.
just a clarification, you would you be the person who would be taking in this Saturdays like
Yes,
frequently?
yes, yes.
Okay, okay.
I'll be taking the 3 to 5 classes on Saturdays. I think there is also a class on Monday by another TA, another PMRF TA. So that is I mean, I
Yeah,
think,
yeah. Monday, I guess I attended few classes.
okay. No, Saturday classes I have been taking so far. I have taken I mean, I've taken all weeks so far, and I'll be
Yeah,
continuing to
today
do
I guess I
so.
missed most of the classes. I started listening from that epilepsy thing. So it
Okay.
was good. Thank you for the class. Thank you,
Thanks,
ma'am.
sir. You will be able to access the recording of the class. I'll be putting it up in the problem solving sheet. You can access that from there, and I'll also be putting up
Okay.
the slide also. And yeah,
So, how do is it going to be sent to by mail or
no, there
in the
is
account?
no, no. So if you go to the portal where you access the weekly lecture videos, right? At the very bottom,
Yeah, got it.
so you will find the wait. I'll shoot. At the very bottom, you will find this problem solving session. So there you will find if you go to this particular one, you will find the uh, YouTube Yeah, if you go here, you will find the YouTube links and I'll also be sharing the presentation folder link over here. So these are the ones I'll be updating weekly.
So all these
So these are the YouTube
sessions are being
videos.
recorded.
Yes, these sessions are being recorded. You will find the YouTube video links for the same, and you'll also get access to the slides that I am discussing.
Okay. Thank you. Thank you so much.
Thank you, team. So, with that, if no one has any further questions, I'll close the meeting. Thank you, everyone, very, very much for attending. Hope you have a good weekend.